Thank you very much. Thank you for organizing this. This is a very big deal for us, and I want to thank ELIXIR for providing motivation and all the logistics for this event. This is an introduction. This is the first webinar in the series, and so we're not going to be talking about specific analysis here. We're just going to talk about philosophy of why we're doing this and introducing some key points. We're gonna more or less stick to these points. I'm gonna give like a brief introduction of how the coronavirus works. It's a beautiful but deadly machinery. Then Frederik will introduce the Galaxy project for those of you who may be not familiar with it. Then I'll again explain what we're trying to achieve here and introduce some of the genomic analyses. And then Sergei will take over to talk about evolutionary analysis, and Björn will wrap up with chemoinformatics, viral drug screening types of analysis. Again, our goal here, as you all know, coronavirus has no boundaries, doesn't have any political ideologies, it's just everywhere. So our main motivation for starting this was to mount a public open effort to analyze SARS-CoV-2 data, coronavirus data. And so this is how this evolved. And the URL for this resource is covid19.galaxyproject.org. It's a resource which is maintained by the worldwide Galaxy community Frederik will introduce. So just a brief introduction about coronavirus. It's a very large positive strand RNA virus. Positive strand means it's a ready transcript which gets transcribed in the human cells. It's very large. It's more or less, give or take, 30,000 base pairs, and it contains several protein coding regions. Actually contains quite a few protein coding regions, which are shown here. So the first 2/3 of the genome, these very large ORF1A and ORF1B, which encode for so-called non-structural proteins, and that is followed by structural proteins such as S, the spike protein, which gives coronavirus its appearance, and other proteins such as the envelope protein, membrane protein, nucleocapsid protein, and some auxiliary proteins. And all of this gets assembled into this particle, which you see. So it's a one very large mRNA. It has a cap, it has poly A tail, so it looks like any other human or mammalian mRNA. It has a very interesting way to transcribe itself. So it's a protein which encodes its own RNA-dependent RNA polymerase. It's a positive strand RNA. So it gets transcribed into negative strand. From negative strand, you again generate positive strand transcripts. And what this picture shows is that you have one long transcript, but you also have this nested shorted transcript, so-called subgenomic RNAs. They all share the same cap. This is because when the new positive strand RNAs are made, they're made from this end. Each protein coding region contains so-called these TRS sequences, which are complementary to the leader sequence. And so what happens is that when RNA produces this transcript, it jumps and incorporates essentially it performs template switching, and it incorporates the header, the 5' region. So despite the fact that all these RNAs have different length, the leader is the same. It's the long RNA has the same leader as all of the other subgenomic DNAs. And you can see that, for example, the longer RNAs, they contain multiple open reading frames, yet only the first one is translated. So by generating this subgenomic RNAs, virus ensures that it makes all the proteins. And another interesting feature of coronavirus is this, you can see that ORF1A and ORF1B overlap, but they're not in frame. So normally ORF1A gets made, but in about 20% of the cases, there's a slippage site here, which there is a particular secondary structure at that region. So the ribosomes slip -1, and they start translating. They essentially switch the frame and start translating in ORF1b frame. So most of the times, just ORF1a is made, but in about 20% of the cases, there is ORF1ab is made, which looks like this. And these transcripts encode proteases, in particular the main protease, or which is this one, 3CL, which excises subproducts from this long polypeptide, and therefore produces all the proteins the coronavirus needs to complete its life cycle. One of the unique things about this is a very large RNA virus, and one of the things that allow coronaviruses to be so large is that they have proofreading activity. So they encode exonuclease as one of the non-structural proteins, and that really increases their fidelity, because normally when we think about RNA viruses, we think about systems which have fairly large, fairly high error rate. So this bar in this picture shows the error rate, so errors per nucleotide per replication cycle, and you can see that addition of that exonuclease bumps it from something around 10 to the -5 to something around 10 to the -7. So coronavirus does not mutate as rapidly as we, for example, as HIV, for example, which is of course a different kind, but also an RNA virus. And this is another slide to emphasize the point that fidelity allows coronavirus to have a large genome. Having large genome allows coronavirus to encode a number of auxiliary proteins, which help coronavirus to evade host defenses. So it's a complex virus with a complex life cycle, and it's to a large extent enabled by the fact that it's replicating fairly accurately. So this is overview of coronavirus molecular biology from 10,000 feet. And at this point, again, our goal was to enable open analysis of existing coronavirus data, and we using Galaxy platform for that, and Frederik will introduce what that is. Frederik?
Okay, so will you drive the slides, Anton?
I will, if that's okay, yeah.
Yeah, perfect. So I assume most of you know Galaxy already as a project, but in case not, Galaxy is an open web-based platform for accessible, reproducible, and transparent research. It started in the biomedical field, but now a lot of different communities are working on this. And this has been truly a worldwide community. So on the next slide, you can see that it started in the US and with the main site started by Anton and James about 15 years ago, but has evolved to a truly global effort with a lot of development going on in Europe and Australia, but also in Africa, India, Asia. You have large communities working on Galaxy. In the next slide, please. This is has led to really an enormous amount of public galaxies available for domain-specific research, and we have over 100 platforms, and we have hundreds of developers and contributors across the world. Also within ELIXIR, a lot of the nodes have activities in this, and that is why we started about 4 or 5 years ago a ELIXIR Galaxy community. On the next slide, you can see that this is really encompassing all the aspects that we do in ELIXIR. We try to integrate from data, compute platform to training, and we try to serve the different communities that we have that are more oriented towards research domains such as proteomics or plant sciences. And all of these researchers are in a lot of cases using Galaxy as one of their primary platforms for data analysis. And not only within ELIXIR, this is relevant, but this is also been driving a lot of the efforts on the technology side. Within Elixir, you have the tools platform, and we are now developing a new ecosystem around tools and workflows where Galaxy also plays its part. And for example, we have now recently fast-tracked the workflow hub, where also the Galaxy workflows that will be described in this series are already registered. But also EOSC-Life, which is the European project to drive the European Open Science Cloud in life sciences, has a strong activity in Galaxy, and a lot of the technical components underlying Galaxy are shared with what we want to do in EOSC-Life. On the US side, you have the AnVIL project, and this has similar goals as EOSC-Life in a way. It is about inverting the model of genomic data sharing. Rather than every researcher setting up its own environment to do the analysis, we want to move this towards bringing the researcher to the data. That's an important role there. This is an NIH funded project on the US side. So what does Galaxy look like? In case you aren't familiar, this is the interface. It's a web-based platform, so you run it through the browser, and you will typically get this view where on the left you have a long list of tools. On the right, you get your data sets that you are working with, and in the middle, you get an interface where you can select parameters for your tools, run workflows, or build your workflows. This is standing on a whole ecosystem. We currently have in the Galaxy toolshed over 7,000 tools available that can be integrated in a Galaxy instance. We have been working on shared reference data. We use a distributed file system to make sure that across Galaxy instance we can have the same reference data available. This is, for example, the genome sequences that we are using in this. Also, to make development easier, to facilitate contributions across the world, we have several development kits to make new tools available in Galaxy, to install reference genomes in your Galaxy instance, to install the Galaxy instance itself, etc., etc. Also for bioinformaticians, we have an API, so you have programmatic access, so you can easily automate all the analysis and integrate this in pipelines. Because of course, this is a rather complex system now. We have spent a lot of time in training materials, and this is now collected all at training.galaxyproject.org. You will find over 150 tutorials there, both on the kind of research aspects, so for the different domains, but also on how to contribute to Galaxy or how to develop for Galaxy. We also already have 2 tutorials for these available at the place. And foremost, we have a lot of features for sharing data, sharing workflow, sharing analysis, which is really why we are using Galaxy, because this aspect really facilitates the sharing between researchers across the globe. You can find all the information on galaxyproject.org. And one important aspect that I want to highlight, I don't have the time to go into all the details and features of Galaxy, is the fact that you can string tools together into workflows to make sure that you can reproduce your analysis easily with all the correct parameters, tool versions, etc. And you can share this with colleagues across the world in an easy way. And we can do this for these asynchronous workflows that you just run and wait until the result is there. But we also now have synchronous workflows available, which are, for example, Jupyter Notebooks or RStudio, which we can directly run from the Galaxy interface, and where also data that is available in Galaxy can be used within these environments. This is for some of the visualization, for example, used in these COVID workflows. So we want to distribute all of this to the broad community, and we've actually started thinking about how we can streamline this a couple of years ago when we started the useGalaxy.star project. The idea was that we have all these public Galaxy instances, and we want to reinforce the collaboration and make sure that there is a certain alignment between tools, workflows, and reference data available across these instances. Because it really depends on the exact version of the tool, the exact version of the reference data, to be able to reproduce analysis across Galaxy instances. And this is non-trivial. So we've started setting out with a number of people in the Galaxy community to develop this. And this has helped us tremendously in now bringing this COVID work to different instances across the globe. Because in a few days after we were contacted by Anton if we could deploy these workflows also on some of the European galaxies. We have been able to do this, and also on the on the Australian Galaxy side. So currently we have at least 5 Galaxy instances which would have these workflows available, and where you can reproducibly run these analyses in the local resources. And to make sure that we have sufficient compute resources available, we have also implemented distributed computing inside Galaxy. So this is a so-called pulsar network. And for this, we focused on the useGalaxy.eu, which is run by Björn from Freiburg. And this Galaxy instance can not only use the local resources of the German cloud, the Denby cloud, but can also submit jobs to various locations in Europe. And we have now, I think, almost a dozen locations available where from the useGalaxy.eu website, analysis can be run across the globe without the user even needing to know that this is the case. And this has allowed us very much to expand the number of resources available for users, specifically in this time where a lot of people want to reanalyze data using useGalaxy. And this is really an easy way to scale up and not be limited by local resources. And as you can see, we have also made a link with Australia, where we in this case send compute heavy jobs rather than data heavy jobs because of the bandwidth, of course. All of this preparatory work that we've been doing with the Galaxy community in the past years has allowed us to deploy the COVID workflows in a very rapid way and in very diverse ways. So we have the different useGalaxy instances where these are available. Also, Elixir Italy has deployed Laniakea, which is a service that allows you to run different Galaxy flavors on demand. And the COVID one is one of these flavors that they've added. If you want, you can also deploy this locally on your own computer or your own cloud. We have made containers available that contain everything that you need to run these analyses. Here. This is, for example, also useful if you need to run this on sensitive data within your firewall. You can set this up, this exact same environment easily locally. And if you have already an existing instance, then we made it easy to install all the workflows, the tools, reference data, etc., through automated procedures. And you can find all the data on the COVID-19 Galaxy project website, where this is detailed, and we have some instructions there how to go about that. Thank you. Back to you.
So now I'm going to briefly explain what we're trying to do here. So data analysis is basically like cooking. You need good ingredients. That's usually, well, you need ingredients. That's usually data. And then you do things with these ingredients. And in case of analysis, you do them with tools that analyze these data. So you don't have a stove, you have tools. So what we can control as a Galaxy project, we can control tools. In other words, we can provide access, free access to open source tools using public compute infrastructure. So our goal really is to enable anybody in the world to go to a platform provided by Galaxy, use any tool they wish to use, and ensure that this tool executes on small data set or large data set, because we not only provide tools, we not only provide workflows, we also provide the infrastructure. And I guess our hope is that given the set of ingredients, we can make, you know, food which tastes consistently good in terms of what sort of final analysis is. So the problem with the general problem with the coronavirus right now is that despite what you might read in New York Times or Guardian or, you know, Figaro, is that in fact the state of scientific state of SARS-CoV-2 analysis is not so great. And again, as I said, there are two components to cooking: ingredients and how you do it. And in terms of data analysis, that's the data and how you analyze the data. So let's talk about first how people analyze. This is a table from our bioRxiv submission, which is now which will be published in PLOS Pathogens in short order. This is a set of tools that the first 4 papers reporting genomic analysis of SARS-CoV-2 used. So you can see that, well, there are some similarities, but for example, paper number 4 here uses something completely different. And in fact, that paper cited Freebase, a well-known variant analysis. So it's a tool for which calls find SNPs. So that paper listed it as an assembler. So apparently they did assembly using Freebase. So it's the kinds of the things that we are trying to avoid. If you provide scientific community with a well-documented set of latest tools which are suitable for this kind of data, hopefully we will prevent using Freebase from being used for assembly. So our first goal is to enable that analysis that are conducted are reproducible, because this was sort of a philosophical goal of Galaxy for the past 15 years. Another problem is, of course, is the ingredients, the data. So the situation with SARS-CoV-2 data is really bad. At this point, we have about close to 900 raw read data sets, the actual reads, Illumina reads, Oxford Nanopore reads, and also some BGI sequencing machine reads. And this is a mixture, again, some Illumina, some Oxford Nanopore. Most of the Oxford Nanopore data is in processed form. So these are FASTQ data sets where bases have already been called. So there is no underlying files with actual raw electrical signal. And these files are absolutely essential for many downstream analysis, such as, for example, variant calling, proper identification of indels, various types of expression analysis. You cannot really use Oxford Nanopore data without having access to that primary data. Another paradox is that most of the complete assemblies of SARS-CoV-2 go to GZ, which is a database. And so there are, well, soon probably going to be 20,000 sequences there. But remember, there's only 900 raw read data sets. So where is all this data? How can we ensure that these assemblies are actually accurate without having access to the primary read data? And there is, well, we'll show some of the interesting examples. So it's really, unfortunately, the state of affairs with the COVID-19 research, it's not open at all. People are hiding data, and accessing to even supposedly open data is not that easy. For example, GZ is a great example, is that it's supposed to be an open database, but in fact, you need to get an account in order to access this data. You cannot redistribute this data. So for example, just imagine you're teaching a class and you want to show people, for example, how to estimate, how to date most recent common ancestor. You cannot use this data for the class because that violates the data use policy, which I have on this slide. Plus, it's very difficult to get data of GZ. So the whole idea of GZ is very unclear to any of us. And the open databases, for example, the GeneBank or whatever NCBI runs, have only a subset of these 12,000 complete genomes. And this is a wonderful example. Sergei, maybe go ahead, describe that.
Oh, yeah, sure. So one of the expectations of you know, a database service provider is that you have an understanding of when the record was deposited and when it was edited. And one of the things that we discovered during our daily analyses of all the genomic sequence data is that sometimes records get edited without leaving any trace. So for example, just to give you one, there's a specific sequence that broke our scripts because it contained the literal word virus in the middle of its nucleotide sequence, which was, you know, clearly an artifact. And it was deposited there, and as April 2nd, it was there. On April 10th, it disappeared. So if you download the data later, you know, it's no longer there. However, the record has never been updated, and it only shows that it was edited on the 27th of March. So things just change, and you have no way of knowing that they did unless you took care of versioning yourself.
So it's an example where there is no any version control either on the GZ8 side. Well, there are some other issues with this. Is now an example of short read data. This is a data set which was deposited to short read archive, and you're sort of trying to figure out what this is. So on one hand, it says that it's an anal swab from a bat, but if you read the description, and it says that total RNA extracted from the bronchoalveolar lavage fluid, so from the lungs, which is so it's not quite clear what is where. So in view of these strange things, our goal is to provide uniform access to latest tools and workflows so people use the same set of tools. Again, Galaxy provides thousands of tools, so we're not imposing a particular best practice workflow. We suggest some workflows, but people are free to experiment. But at least they'll be experimenting with the tools which are properly versioned in a standardized environment. This will also ensure reproducibility and transparency on these analyses. As I mentioned, we suggest best practices, but again, you can experiment. But that experimentation will be documented, because once you share the analysis you perform in Galaxy, it's known which versions, which parameters, all that information is known. And again, because we provide it on infrastructure, you will be able to run small analysis. You will also be able to run large analysis thanks to a great public cyber infrastructure, both here in the United States and in Europe and in Australia. And as you will see today, at this point, we broadly divided our analysis in three groups, so genomics, evolution, and chemoinformatics. And I'm going to briefly introduce our genomic analysis. So there are several kinds of them. I'm just going to highlight two. One of them is assembly. Assembly of SARS-CoV-2 is not particularly complicated if you have good. It's a single short transcript. The complication there is that the data is usually metatranscriptomic in nature, because you sequence, for example, the content of the fluid from the lungs. So you're sequencing everything. There are human reads, obviously. There are any reads coming from secondary infections, and there are SARS-CoV-2 reads as well. So the workflow we provide uses Unicycler. It's a spades-based assembler, but there are other assemblies in Galaxy if you want to try something else. Although this particular workflow gives very good results. The subject of the next webinar next week will be on variation analysis. And variation analysis is peculiar for these data, because most of the software, most of the good publication tested software is geared toward analysis of human data, so diploid samples. Here it's a haploid mixture, so genome is small, coverage can be very high, and when you have very high coverage of very small genome, even if you're doing it with a platform which has low error rates such as Illumina, eventually you're going to have every site variable. You're going to have variants at every. So how do you decide which ones are good and which ones are not so good? So that requires a certain logic, because again, it's a special case of variant calling, and we're going to dedicate the entire webinar to that problem. how to properly call variants in viral mixtures. And this is some of the results which of course require interpretation, and Sergei will talk about this interpretation, biological insights that you can get out of this. But this is map of variation across the genome. The different colors here represent different samples, and the shapes, for example, triangles are non-synonymous changes. Those are the changes that change underlying amino acids. So circles are synonymous changes, and you can see that there is a lot of dust, there is a lot of small, low frequency variants, there is a lot of sites which are fixed, yet there is also a lot of sites with intermediary variation. We still don't know what the extent of intra-host variation in SARS-CoV-2, how much variability you have inside a single patient. Are there differences between sites, for example, lower respiratory tract and upper respiratory tract? Is there evidence for multiple infections? Is there evidence of recombination? So it's important to know variation within samples. And as I mentioned, the current available data doesn't really allow for that, because we have thousands of complete genomes, but only few primary data sets. So there are other workflows which are already exist for read processing, for timing of most recent common ancestor, for analysis, recombination, and selection. Also, we're working on developing Oxford nanopore-centric analysis, which will work with FAST5, the raw data, and also in collaboration with Sergei, we're working on a comprehensive evolutionary analysis, which would perform which will allow to gain multiple types of insights. So Sergei now will talk about what kind of insights.
All right, I'll tell you briefly about the work that we've been doing on studying viral evolution and looking for evidence of natural selection in this pathogen. And there'll be a separate seminar dedicated to this entire topic, I think on May 20th. So despite what you might have heard or read, there were other viruses before SARS-CoV-2. So a lot of the scientific research reads like this is the first time we've ever seen this. As Anton as mentioned, most RNA viruses actually mutate faster than SARS-CoV-2 because they lack proofreading exonucleases. And just because you see a mutation or even a GASP non-synonymous mutation, it doesn't mean that the virus is evolving to become more pathogenic. Therefore, I find it really disturbing when what I consider to be a reputable publication when I was a kid, like Newsweek, publishes things like first significant coronavirus mutation discovered in a preliminary study, and that mutation occurred exactly once in all of the observed 15,000 sequences. Next slide, please. So coronaviruses have been around a long time. In fact, before it was fashionable, we published a study that estimated that the progenitors of beta coronaviruses, which is where SARS-CoV-2 lies, diverged from their closest relatives like alpha coronaviruses and avian viruses 200 million years ago. Next slide, please. For the conspiracy theory minded, everything related to coronavirus research is very prominent. So if you look at the access record to the paper that I just cited, obviously see a spike in 2020, but curiously we also see a spike in October, in late 2019. I have no idea why, but what did these people know that we didn't know? Next slide, please. So it is important to understand that viruses evolve initially within a specific host. So within a specific human or bat or anything that they might infect. So viruses have error-prone replication, and in the case of SARS or coronaviruses in general, they also like to recombine. These processes generate variants that exist in a specific host as a part of viral quasi-species. So quasi-species is basically a term that's been adopted originally in HIV research to describe the fact that viruses that exist, some RNA viruses that exist in specific individuals are not homogeneous. They actually act like populations. And as such, they're subject to rules of population genetics. So you have an evolving population of individual viruses that have different fitnesses, generally very similar, and they'll be subject to things like genetic drift, selection, and migration. And one of the benefits of using raw read deep sequencing data is that you can actually estimate and quantify what's happening within specific hosts. So next slide, please. So here's just a very small, very high level plot of what happens within a specific short read archive accession number that's referenced here. So this is just a plot of differences from the reference genome that we observe in this particular sample. This is using the pipeline that Anton had described within Galaxy to covariance. So this is a boring sample. There's a single basically viral population that has 1, 2, 3, 4, 5, 6, 7, 8 differences from consensus. So there's nothing happening in this individual. It's all homogeneous. Next slide, please. You can start looking at other variants, and you see there are now blips that occur at low frequencies that are highlighted by an arrow, and they're private, which means that they're not seen in any other isolates. So they just probably are unique to that individual and never got transmitted anywhere else, which is what you would expect most variants to do. Next slide, please. And finally, get to these interesting cases where this is again a plot of variants within a specific individual, but now you see two mixed populations, one at high frequency with 5 tall bars, and another at lower frequency, but some of those bars are in solid colors, which means that they were also observed in other individuals. So that might look like an infection with multiple strains in the same host, which would indicate that this particular individual now has an evolutionary opportunity for the viruses to recombine. And I would like to emphasize that these types of insights are impossible to gain if all you have are assembled genomes, because that information is by necessity lost since everything is represented by the majority strain. Next slide, please.
Hello.
Yeah, there we go. All right. So once evolution within a host has done its thing, the virus has to transmit to the next host. And has been studied for many, many years in other viruses, especially things like HIV. The different hosts impose generally different selective environments. So upon transmission, a virus that was successful in the previous host, for example, due to an immune escape mutation, may become unfit because that mutation is no longer beneficial in the new host. This is create complex evolutionary dynamics, which have been characterized and studied in depth for other prominent viruses. So, for example, in the picture on the right, you have an image from a paper that we wrote back in 2007 to explain what might happen within a specific individual. So, for example, you can have an individual in the top bar that doesn't have a selective allele, so it's missing a specific immune allele that might cause the virus to mutate. So it does nothing in that individual. Then it moves on to the next individual, which is now HLA plus. There it develops and fixes a mutation, which is then transmitted to another positive individual. If it's then transmitted to a negative individual, then the mutation will revert and so on. But this assumes that you have enough time for the virus to do all of this within an individual. And the bottom panel shows you what happens if it's partially resolved. So if it's sort of on the way up, a fit variant may be transmitted or an unfit variant may be transmitted. So these types of situations occur often, and they create complex mutational patterns that we might be able to exploit. So viral populations also undergo severe bottleneck events during transmission, where only one or a few of many possible particles actually goes on to establish new infections. So you have boom and bust cycles of genetic diversity, so to speak. Next slide, please. There are major selective forces that we expect to act on the virus in short time scales, and this is important. I'm not going to talk about what happens in species or epochal time scales, because those also matter, but we don't really care about them here. So the most intuitive one would be drug-induced selective pressure. There are numerous examples, literature like AZT for HIV-1, amantadine for influenza A virus. And these are likely not currently relevant for SARS-CoV-2 simply because there are no large-scale antiviral treatments being deployed in the population. The other type of selection we might expect to see is escape from antibody-mediated immunity or vaccines. This is manifested in things like antigenic drift and shift in influenza, typically results in changes at viral epitopes targeted by the response. Because there's currently no vaccine at scale, there's some trials, but they're too small to matter at population level, and no previous population exposure to similar viruses. This implies there is a lack of cross-reactive immune response and likely of immune-mediated pressure. We'll see this, but we'll see this later as the infection starts cycling through the population, and that's what it ends up doing. You can also have escape from other types of immunity. So CTL immunity, so cytotoxic T lymphocytes can act quickly enough to matter. It will be a population-specific selection, so that's something we might be able to see now. And you'll also have other selective forces following zoonoses, so fine-tuning to new hosts for things like replication efficiency, better receptor usage, and whatnot. These are usually difficult to untangle until much later on. Next slide, please. Selective signatures can be, natural selection can be inferred, and this is the key word here. It's a statistical statement from substitution patterns or other features of sequences. So there are basically three key lines of evidence that we use to try to analyze what's happening with SARS-CoV-2. So if we use standard technique that we've spent a lot of time, developing and testing in other living organisms, so the rates of synonymous or silent and non-synonymous or missense mutations can be inferred and compared through the quantity called DNDS to detect either purifying, which is subject to functional constraint, or diversifying positive selection, for example, immune-driven. We can also look for frequency changes over time to identify selective sweeps that can indicate directional evolution. This is something you would expect, for example, in drug resistance type situation. Comparing with related, and finally, because SARS-CoV-2, as I mentioned, is an ancient virus, we have a lot of its relatives sequenced and sampled, so we can compare the evolution of this particular system to other beta coronaviruses to help us put what it's doing in context. Next slide, please. There's an unprecedented rate of data generation, as Anton mentioned. So, and this is already out to date. So yesterday there were 12,000 sequences, or nearly 13,000. Today it's nearly 16,000 sequences. We actually have a live sort of tracker of all the genomic sequences versus all the diagnosed cases broken down by country and data deposition. This is work due to Stephen Weaver, who's in my lab, and it's linked at the bottom of the page. But basically, you can see that at the moment, the pace of sequencing is kind of keeping up with the rate of total confirmed cases. But we've sequenced a very small fraction of it. So there's about 3 million confirmed cases, and we have about say in the order of 10,000 sequences. But it's unprecedented compared to other viruses. So this is a lot and lots of data. Next slide, please. So as a teaser for what I'm going to tell you about in a couple of weeks, is there evidence of selection in SARS-CoV-2 genomes? The short answer is yes. We have the project URL, COVID-19 galaxyproject.org. You can see our data of what's going on. And just to give you a sense, there's a chart, again, of basically a very large, very sort of coarse genomic annotation. And we have categories which can indicate that something interesting is happening at the site. And you can kind of see that, there's a lot of signal. There's a lot of signal across the genome. The important question is, if we find some statistical evidence of selection, is there any evidence of functional impact of this selection? And the answer to this is we don't know yet. And most importantly, don't trust anyone who tells you that they, at the moment, it's premature at best to talk about functional impact of any particular mutation. And at the surveillance and hypothesis generation stage, so we're trying to filter signal from the noise. Just to give you a data point, with the 13,000 sequences that we have, 60% of the entire SARS-CoV-2 genome has a mutation in at least one isolate. And as we continue sequencing data will eventually reach the coverage of 100%, presumably. So clearly not the entire genome is subject to selection. That would violate all kinds of things like the neutral theory. Next slide, please. So on our project page, we're basically provide ranks of individual sites. So here's the top 5 sites as of the last analysis. It's not that important, what those things are. I'll explain them to you in a second. All I would like to point out is that none of these sites come from the spike protein, which is the prime suspect for immune escape. They're, in fact, all but one come from non-structural proteins that Anton described early on. For example, RNA-dependent RNA polymerase and NSP2, which has cryptic function that we don't, as far as I understand, we don't know exactly what this protein does, and the helicase protein, and there's one from ORF3A. Next slide, please. So how do we call sites? Again, this is just a very, very high level overview. So we will look for statistical tests. We estimate these DNDS quantities and look for either excess of non-synonymous changes, which would indicate positive selection, or the non-synonymous changes would indicate purifying selection. Ignore evolution along terminal tree branches. Help us if sequencing there's dead end mutations and kind of remove focus from intrahost evolution. And one of the things we require for a substitution to be interesting is that it occurs multiple times in the tree. So for example, here on the right, there's a tree one side in the OR1A, just highlighted by arrows. There are 3 independent introductions of one specific substitution in this tree. So it's kind of a repeated event. So evolutionary replication, if you will. Next slide, please. We can also just look at frequency trends over time. We have a chart relative of 2 residues broken down by day of sampling. So the histogram is how many sequences were depleted and adjusted at any particular day. In this case is an alanine. It was basically, it's the only thing that we need until March, and then March we started getting blips. And it's sort of kind of bubbling around at low level of frequency, except with a spike in early April. So we can look at time series like this and basically look for whether or not the frequency of a particular residue changes over time. And if it does, that's another indication that something might be happening at the site. Next slide, please. We also look for evidence. We link the 2 types of analyses. So for example, for helicase 504, the popover that you see displayed are all the deep sequencing data sets that show minor allele frequency, this population at this site. That means that not only do we see fixed differences between genomes, but we also see evidence that within relations are doing something at that site, which is basically required for any change to be evolutionarily meaningful. And next slide. And finally, we can look for at other beta coronaviruses and ask the question, does the evolution of related beta coronaviruses predict observed residues at SARS-CoV-2? For example, in SARS-CoV-2, at site 97 and reverse RNA-dependent RNA polymerase, the consensus residue is an alanine, and there's a minor variant of a valine. In alignment of related beta coronaviruses, we only find synonymous variants of alanine. We have a model-based statistical imputation technique that suggests that the only expected residues are codons that code for alanine, so GC anything. And the presence of valine is unexpected based on what other beta coronaviruses have done in the past, could indicate a different selective pressure or new evolutionary path with this site. Next slide, please. But for most sites, actually the opposite happens. So we don't find anything evolutionarily unexpected. So for example, at site 63 in our DRP, the SARS-CoV-2 consensus is D with a minor variant of Y. Alignment of related beta coronaviruses, we find synonymous variants of G and a low frequency of D. Model-based imputation now suggests that while G and G are the 2 most expected amino acids, Y is also predicted to be a credible evolutionary outcome because the model extrapolates evolutionary propensities beyond observed residues. So in this case, let's say, yeah, all right, Y is not too unexpected based on what we saw with related coronaviruses. And I think that's all I have. I'll pass it on to Björn.
Thank you. Björn?
Yes. Next slide, please. So we are leaving now the genomic area, and we are talking about another great piece of infrastructure that was built previously and is now ready to use. And we are talking here about the diamond light source. So one of our partners, so the Diamond Group or the XChem group from Diamond, they used this national synchrotron from the UK to actually create the crystal structures of one of the main proteases that Anton mentioned in the genomic part. So we have the 3D model of this protease now, thanks to Diamond. And what we can now do is we can try to design and drug candidates. So the question is still, can we treat COVID-19 with a new drug, with a drug that is already known, and how can we figure that out? And Diamond was essential for that because they did the crystal structure, so the 3D structure of this protein, and they did also a fragment screen to that. So they took very small molecules, they observed how these very small molecules bind to this protein, and this data they provided for the entire community for free. So the next step is that what we did is we built a community effort around that. We took this data and now we designed workflows around that that try to predict the best molecules that we then can later test. So this all happens in silico, so inside a computer. And what we are doing here is we take the initial fragment hits, we generate enumerations, so we generate charge enumerations, we generate 3D structures of potential drug candidates, and then we dock them. So we simulate the docking of the small molecule with the crystal structure, with the hope that in the end we can have a huge library with potential drug candidates. And this is one workflow that will be presented in 2 weeks by Tim Dutchin and Simon Bray. And they will talk more about this entire project in more detail. But essentially what we did, we screened over 60,000 compounds. We generated all different poses. We docked them into the structure. And in the end, we also try to classify them, we score them, so that we hopefully come up with a list of the top 100, top 500, top 1,000. And these compounds we will then buy and test it at first in vitro. And if the experiments look good, we can go for in vivo. What you're seeing here is that Galaxy can handle millions of docking poses. You see here a history list. So every column which you see here corresponds to a Galaxy history, which runs more or less one experiment. They are all running at the same time. And you, in the other figure, you see one of these figures of the protease with a docked compound. And this can be done in Galaxy. Interestingly, what we used here is resources from across Europe. So what Frederik was telling you before, that we actually can do the docking in Finland and the scoring because they use GPUs, we do them in the UK. This is actually possible right now with infrastructure that we built the last years. This is what we used to achieve that, and we were able to score that in a week. So from the start of the project, when Frank and Tim's team contacted us, we were able to get that running, the first results in over a week, with all the infrastructure that we built the years before. Next slide, please. Just as a small outlook, that was a pitch more or less for in 2 weeks. So you will get many more details, but time is running. So I would like to thank the entire team that was involved here. And this is, as the topics are super diverse, the team is super diverse. So we have assembled experts from around the world that involves Australia, US, Europe. There were a lot of funding involved. You can imagine if we talk about the Diamond light source, but we also talk about huge cloud environments, HPC computing, funded by US government, funded by the European government, funded by the Australian government. And we would really like to thank all of them and also the researchers that put their brain in, their tools in. All this is just possible because we can rely on the Bioconda community, on Galaxy community, on people that provide cloud access and develop all these super cool algorithms which we then can use. So without them, this wouldn't be possible in this fast way. But I think one keystone we would, well, one key point we would like to do, what we like to pitch here is, because we have all of that and we carefully created that and reviewed that before, we were able to use it really fast in a week of time to create actually these results and keep going. So investment in infrastructure is probably worthwhile, as you see here, because this is what we rely on. So thanks for listening, I think. Thanks, Anton. Thanks, Frederik. Thanks, Sergei. Keep in mind, this was just a teaser. So next week, Anton will go way more into detail about variant calling. Then will the drug design part by Simon and Tim, and Sergei will be in, I think, 3 weeks. And we will conclude with a more technical one driven by Marco from Italy and Gian Mauro about the entire infrastructure that we have built if you are more the techie part. Thanks a lot for staying with us.